Okay, good morning. So, Wednesday, the 19th of January, and so we're continuing in our cruise through Chapter 1, and if I get my act together, we'll get into Chapter 2, which is about studying DNA, and then we'll call it a day. Just as an announcement, Friday's good to go, but I may not have a live stream. I'm working on a scheduling conflict, so check Canvas for announcements, and I'll keep you up to speed on that one. Okay? So, off we go. So where we left off, so I'm backing up just a little bit. I'm again, the point here for the semester is what's in your genome and why is it there. So there's just a tremendous amount of research going on right now trying to figure out, what's in your genome and what's it doing there and what's its purpose. Totally makes sense. And so, again, after the Human Genome Project was declared finished, which, by the way, at some level it's never going to be finished, right? Because the end result of the Human Genome Project was the publication of the reference genome. Certainly not a large database with, even a fraction of the population has had their genome sequenced in its entirety. So, in that sense, the Human Genome Project continues, but it's going to, of course, be tackling different questions rather than just trying to come up with a definitive reference genome anymore. So the Encode was the next iteration of a large-scale research effort. And so one of the things that they did simply was they developed probes that were associated with all of the RNA that they could find that was expressed in a cell. And then they simply, by adding the probes, then we're asking the simple question, well, where in the genome do you find these things? So the RNA, of course, is a result of transcription. That's where it comes from. Okay? And so this figure A in particular, is the one that I want you to be able to sort of tease apart and talk about more than just simply, hoping for a multiple choice question. So one of the things that I can ask you to do is to explain what this picture is showing and what fundamental concept does it challenge. Okay, so what's the fundamental challenge? Some of these transcripts are weird in the sense that they're not what we would have predicted based on our concept of the idea that information flows DNA to RNA to protein, and so therefore at any level we can look at the sequence and we could predict either the level below it or the level above it. So if we have an RNA sequence, we could predict where it comes from, right, in the genome in terms of its DNA. And if it's a protein, we could look at the sequence and we could then predict what did the RNA look like, and therefore what did the DNA look like. And so this is the gene concept of the elements in the genome. The products of transcription are things, sequences that hold biological information. And so the idea then was that, one functional product traces back to a single location in the genome, right? And so that's the principle of collinearity, that from protein sequence to RNA sequence to DNA sequence, you would see roughly a one-to-one mapping, all right? And so some of these transcripts violate the principle of collinearity. They're coming from different regions in the genome as if they're one product. Okay, so we mentioned what the significance of the Encode project was, and in the first of it, of course, is that it developed an enormous database of DNA elements. The second is they've challenged our interpretation of what a gene actually is. Now, the issue isn't resolved, but that's certainly a significant contribution. We do need to explain the presence of those, what I'm calling weird transcripts. Okay? Right? So this is the last part here, is that the challenge from Encode is about function. How do we define function, and how do we recognize functional sequences when we stumble upon them? Okay? All right, so in chapter 2 of your book, it leads off with this sentence. And so I wanted you to kind of to see this. And what I'm hoping that your reaction to this is that, well, we must be missing something here, because I don't think he really means what he seems to be saying, because he's basically saying virtually everything we know about genomes and genome expression has been discovered by scientific research. Theoretical studies have played little role in this or any other area of molecular and cell biology. And it's like, man, those are fighting words. First of all, I would say, well, theoretical studies are very much part of scientific research, so in that sense, that statement's falsifiable right off the top. But my other point, well, it's a minor one, but this idea of genome expression, that's not a settled fact either, because it's genes that are being transcribed. The genome is a dynamic feature of organisms in the sense that the chromosomes are being packed and unpacked, but of course the goal is not to express the chromosome. The goal is to express a handful of genes because some environmental signal has suggested or demanded that the cell responds. And so gene expression is a response to cellular signals. So, I didn't like the phrase genome expression, but that's a debatable one, right? It's sort of an active area of research, if that's an appropriate way to talk about it. And certainly theoretical studies fit well into what you would call scientific research. So in that sense, it's a horrible sentence. But I do think he means something more than that, and so we'll kind of tackle that a little bit throughout the semester, this idea of what are theoretical studies in the sense of how do they contribute to our understanding of what's in the genome. And so the reason I responded so was because, of course, evolutionary theory is essential to understanding why our genome is the way it is. It has a history. And if you don't appreciate that phrase, then all you're doing is collecting, and then you're postulating why is it there, but ignoring the fact that there's a history to those elements. Okay, so that's partly why I'm introducing a little bit more about evolutionary biology right up front, because I want you to appreciate that, of course, everything in your genome has a history. You're the product of uninterrupted ancestry going back before history. So at any rate, these are fun statements, because you shouldn't just read through them and go, okay, because these are fighting words, man. All right, so let's get back to the Encode thing, and I added this slide, and I apologize, it's really quite busy, but let's put it this way. The argument, why are those rare transcripts that seem to defy our principle of collinearity, why are they there? And so there are really only two possible arguments once you get over it's just technical error. And the Encode people know what they're doing. These aren't technical errors. They are printing those out and declaring that they exist because of other principles of science research, right? They've replicated, they've ruled out other hypotheses. So they're not errors, they exist. We did note that they're rare in the sense that in a cell you would see very few copies of those odd, weird transcripts compared to the ones that we are used to seeing. Okay, so that is a catch in their presentation. But okay, let's get back. What are the possible reasons why these transcripts can be there? And there's really only two possible reasons. And the one that a number of people certainly favor is the idea, well, they must be functional, and we just simply haven't discovered what their purpose is. Okay? So notice that this is what we would call in biology an ultimate explanation. It means that you're going to have to come up with an evolutionary argument for why that feature is present. Okay, in some level, the how part has already been explained, right? These are transcripts, so they certainly exist. They map to areas of the genome. So in that sense, the mechanism of how do they come about, well, that's there. But the question is, why are they there? Okay, so we're going to return that to a little bit. So the other argument, so one argument is that they're functional, and we're just ignorant about what their purpose is. Okay, well, the other is that they're non-functional. And in my years of teaching to undergraduates, to graduate students, and in conversations with many of my colleagues over the years, there is a resistance to this idea that features that we can see or measure or observe about us don't have a purpose. Function is purpose. It serves to do some work. Okay? But trying to get you to understand that biology functions as good as it needs to be. It doesn't actually strive, if you will, in that teleological sense. Biology doesn't strive for perfection. And I think intuitively you already know that, but there's a barrier to sort of thinking about this, and so we're going to kind of work our way through this just a little bit. But so the point being is that I can explain, well, not me, others can explain the presence of those rare non-collinear transcripts as the product, again, the how, of RNA polymerase makes lots and lots of errors. And so this is called transcriptional noise. And so I've put a couple of references online for you, and this one in particular. So they went through, and this is now a couple of years old, so in terms of genomics research, this is getting a little aged. But basically the point being is that using definitions that people agree with to a certain extent about, what is a gene, what not, they came up with, and this is for the human genome, they came up with about a little over 42,000 genes. Of those, 20,000 are of the protein coding variety, and another 22,000 are of the non-coding, or meaning that the functional product is some RNA that does work, right? And then they found an additional 323,000 transcripts and change, okay, that they could identify function to a certain extent. But on the other hand, there's 30 million additional transcripts that there's no reason to believe that they're functional. Okay? So it's all about the definitions in part. What do you mean by function? And so getting back, let's talk about the transcriptional noise argument. And remember the behavior of RNA polymerase, right? How does it work? Well, it adds RNA nucleotides according to the DNA template. Yes, yes, yes. But how does it know where to start? And so, looking at E. coli, right, we've talked about the sigma factor, which is a subunit of the RNA polymerase. And the purpose of the sigma factor is to bind to a consensus sequence. In eukaryotes, we call it the Tata box, but there's something equivalent about 10 base pairs upstream of the start of the gene, right? A Tata box, right? In the Tata box is just a handful of nucleotides. In the eukaryote Tata box is just 6 nucleotides long, so a 6-mer sequence, right? And if you just think about it, 6 letters out of a genome that's over 3.3 billion base pairs, nucleotides long. Okay, so the chance that you're going to find this sequence just simply by chance is going to be in the hundreds of millions. And we showed you the math about how to do this, right? So, 4 raised to the 6, so the length of the message is 6, and 4 because there's A, T, G, and C, right? Okay, and so, look, anytime there's a Tata box, you would expect sigma factor, if it's nearby, to bind to the Tata box. That's what it does. It's not like there's a brain working here to go, oh, wait, there's no gene associated with that Tata box. So, at least at that level, that's a one way to generate a transcript, because RNA polymerase will start to transcribe. Once it binds to the TATA box, it will start to transcribe. And then at some point it will reach a stop codon or something, and will not stop codon, but a releasing point, and so off it goes, right? And so we can expect to get, in some cases, some really long sequences that don't correspond to any gene whatsoever. Again, it's called transcriptional noise, and so there's going back, decades of research measuring the behavior of RNA polymerase. This is not just a theoretical construct. It's a, as our author would say, it's science research, right? Okay. Although theory predicted it. Sorry. Can't help it. Okay. So we're going to talk about this throughout the semester, because at some level, I say function and you say purpose. And so you say, what's the big deal? And it's like, well, but you have to understand that we're products of history. And so therefore we have features not because they serve us any particular purpose, but simply because we inherited it from our ancestors. And so, classic sense is the various vestigial organs that we have, the appendix being number one. So people have continued to try to argue and use scientific research to find function about the appendix. But the simple answer is that it's simply the leftovers of when our distant ancestors had a cecum, and the cecum was used as part of digestion, and we don't use it anymore. And primates don't use it anymore, et cetera, right? And so the simplest answer is that it's present because biology doesn't strike, a perfect balance. It's simply reduced in size, but it doesn't serve any particular function. The developmental processes of being a mammal mean that you're going to have something like the appendix, a junction between the areas of the digestive tract, right? So at any rate then, knowing that we're products of history, the way you identify a feature of an organism that it has purpose is in the sense that it must do something that increases the likelihood of survival and or reproductive success. That's why you're here. Your ancestors had kids. They survived long enough to have kids. And you are a product of an uninterrupted line of success in that sense, right? And so features of us, I don't know why I'm doing jazz hands here, features of us are shaped by evolutionary processes. Some chance, yes, but also natural selection. If it contributes to fitness, how many kids, survivorship, then that feature will increase in the population from generation to generation. Okay, so that's our new definition of function. It has to be shaped, built, maintained, honed, if you will, by natural selection, which is the force, if you will, of evolutionary theory that produces adaptations. Right? So biological function, it's present because it's an adaptation. Now it turns out in principle it's fairly easy to define these things, but in reality being able to identify this, and as meaning going through scientific research, right, and demonstrating that any particular feature that you can identify has biological function is challenging. I hope you appreciate that. How do you show that a feature is linked to organismal fitness? You got to show that the absence of the feature reduces fitness. The presence of the feature increases fitness. So how do you do that with humans? I hope you understand that you don't. It's illegal. It's unethical. Don't do that. Don't think that way. But of course, that area of research is done across the world in various laboratories on model organisms, right? So comes from theory, yes, but it's well backed by empirical research. And so what your author meant by scientific research is empirical research, experimental research. Switch over to what are the tools that we use in working with DNA that allow us to identify the things that are present. So if you give me a moment, okay, I'm going to have to switch slides, and so behind the scenes, let's see if this is going to work for us. So, okay, hang on just a second. Okay, so you're going to see me switching some inputs here, so the screen may flash a little bit, so just bear with me. Okay, I think that worked. It was, like I said, there's a lot of stuff going on here, and I don't have a producer, so I'm doing what I can, but I'll get better at it. So, all right, so we're moving now into chapter 2 officially. So, what do you mean by studying DNA? Well, this is where the fun gets in. These are the technical aspects. These are the wet lab, and it's like, yeah, but we're generating so much wet lab stuff that you can't do it yourself. So I hate to tell you, those that you are looking forward to loading gels and whatnot, we'll do that a little bit, but you know what you do? You build robots. And so a lot of what you end up doing is working with data analysis. So that's where the fun actually is you get lots and lots of data and start looking, using your algorithms and whatnot, you're looking for patterns. Okay, so in 307 we talked about genetic engineering just a little bit, and so if you recall, genetic engineering is now sort of morphing into a lot of different things, but the idea is basically, the classical idea of genetic engineering is that you're putting DNA molecules together. The DNA molecules can come from the same organism, but maybe they're different parts of the genome, or they can come from different organisms, in which case you're talking about genetic modification, right? So there's really, though, just two important ways of thinking about all of the techniques that you're going to be introduced to. All of the technology follows basically two fundamental principles. So the first one is that nucleotide sequences, whether we're talking about RNA or DNA, of course, the property that makes them manageable, swapping different parts and whatnot, is the fact that if you have complementary sequences, A goes with T, A goes with U, G goes with C, and so on, okay, because of hydrogen bonding opportunities between complementary sequences, the Watson-Crick base pair rules. Okay, so knowing that, it offers up a way of targeting manipulations. You'll know that if you generate two sequences, and if they have complementary ends, you know that if you do a little effort, you're going to get them to come together. This is the central idea of generating probes, right? Probes are oligonucleotides, so they can be somewhere on the order of 70 to 80 nucleotides long, and you attach some sort of chemical that allows you to make the thing visual when it binds to its target. And what do you mean by target? Complementary anti-parallel nucleotide sequences, hydrogen bonding, right? So then the other principle is the fact that there's thousands and thousands and thousands of proteins that recognize specific DNA or RNA sequences, and they bind there. Okay, so for example, all of those transcription factors bind to what? They bind to promoter regions upstream of the start of a gene, proteins, recognizing specific sequences. What's an example of a specific protein recognizing a specific nucleotide sequence? Well, we mentioned sigma factor. What does sigma factor recognize? The Tata box. Okay. All right, so those are principles, and so of understanding the tools of molecular biology. And so one of the things you can expect on your exam is that we're going to start talking about the tools of molecular biology. I'm going to ask you not just how it works in terms of, like, you know, step 1, step 2, step 3 protocols, but what features of molecular biology are those tools focused on, right? So hydrogen bonding or interactions. Okay, so your book chapter 2 begins by just listing and then explaining how they work, enzymes that are used in DNA manipulation. So I thought we would start there. And so this is a figure that sort of summarizes the purposes, the functions, if you will, of the different enzymes. And so we have enzymes that allow us to copy DNA, to cut DNA, or to rearrange DNA, right? Okay. And of course, it isn't restricted to just working with gene sequences. It can be working with any DNA sequence. Okay. So thinking about this, hang on just a moment. Okay. Yes, it's true. My exams are take home. So of course you could just look this stuff up. I'm hoping, of course, that you have enough, you know, curiosity about this to recognize if you want a job with a bachelor's degree, of course you're going to end up working in a lab, a lot of it. And so understanding the toolkit, this should be part of your second nature. So when I talk about polymerases, you should be able to tell me, well, does it work with a template? What are the substrates? Remember, these are enzymes, so they work with substrates. What are the substrates? So the activity, of course, is, well, how does it do its work? And then, of course, what's the end result of the polymerase's action, or the nucleases and the ligases? And then there's a whole series of enzymes that do changes to the ends of nucleic acid sequences, right? So as you're reading through the book, try to think of it in terms of adding entries into these columns. And yeah, the exam will have questions that would interrogate something like this. I'm not going to just ask you to fill in this and then have you submit it, but of course we'll develop questions that work across these different columns. Okay? So we're not going to talk about this now, but your book obviously points out that you know, really important technology is the idea of polymerase chain reaction, and so you should recognize the enzyme that is at the center of polymerase chain reaction, right? Of course, it's a polymerase. Hello, it says right there. Okay, and then the other major part of, you know, manipulating DNA is the idea of cloning, right? Between polymerase chain reaction and cloning, where you insert a DNA sequence into a vector, and then you put the vector, which now has, hopefully, your target sequence, into organisms, bacteria or yeast, and you allow the colonies to grow, and you use some principle of selection to identify the recombinant DNA that you want. But at any rate, at the end result of this, PCR makes millions and millions of copies of the target DNA that you want. Cloning is the other way that we make millions and millions of copies of the DNA we want. So what's the difference in strategy? PCR, small. Cloning, big, in terms of the size of the sequence that we want to amplify. Okay, so we're not going to really talk too much more about the mechanisms of that. So let's just kind of go through the enzymes. So what do DNA polymerases do? They synthesize DNA from existing templates, and the polymerases that are present in organisms, okay, they add from the 5' to 3' direction, and then they proofread. Not all of them, but many of them proofread by backing up and removing a nucleotide if it's been added incorrectly, right? So endonuclease activity means adding, exonuclease activity means removing as part of the proofreading function. Okay, so as you can imagine though, having an exonuclease activity as part of your polymerase isn't necessarily a great tool, because you just start adding a nucleotide and then it backs up and removes the thing. So that's the so-called Kornberg polymerases. All right. So Clonal is a polymerase in which you've basically cleaved some of the subunits and you've retained the endonuclease activity, but you've removed that part of the exonuclease activity that wipes out your work. And then you should know who TAC is. PCR, hello. What's special about TAC is that it's a Kornberg type polymerase, but it can operate at temperatures of like 72 degrees Celsius, whereas Clonal and Kornberg, they're thermal stable at around 37. That's where they do their business. And then above those temperatures, of course, they fall apart. Okay. So this is a little bit about Kornberg, Clonal, sequenase, okay, TAC, reverse transcriptase. So those are the polymerase examples that are talked about in your book. And again, thinking about your table, here's some of the information that you might want to use, right? But I do want, it's just a couple of pages in your book, and I do expect you to kind of dig through that just a little bit and organize it into a table or something so that you can identify that when asked. Okay, so let's get to nucleases. So nucleases are proteins that you find on your skin. They're, of course, present in your cells. And their business is to chop up DNA or RNA. Okay. And then there are two kinds of nucleases. Either they chop up from the ends, so an exonuclease, or they're endonucleases, where they chop up on in some sequence within a DNA strand. Right? And so endonucleases, the examples are REs, and so of course you recognize that acronym, hint hint, restriction enzymes. So this is a figure out of your book. It shows the, if you don't know what an end is and what an inner circle is. So endonuclease, exonuclease, ends, endo within. Okay, internal cuts, external cuts. Table in your book, just a few names, restriction endonuclease. Look, there's like 800 known restriction endonucleases, so thank goodness they didn't give you a huge table. I didn't print the one table or put into a slide the one table that they have, which has, I think, something like about 10 endonucleases, and then it names the sequences that they recognize and then the type of cut that they make. Okay, so your book doesn't talk about these at the moment, but I thought we would mention proteases. So you have nucleases, which chop up nucleic acids, and then of course there are proteases that chop up proteins. And so in a DNA extraction protocol or RNA extraction protocol, one of the things that you'll routinely do is add a protease called proteinase K, which comes from a fungus. And what it does is it cuts proteins wherever there's a serine residue, a serine amino acid in the sequence. Okay? And there's lots of different kinds of proteases. So threonine proteases, they tackle sequences that have threonine in them, cysteine, et cetera, et cetera. And so, interestingly enough, one of the COVID-19 therapies that's just come out, Pfizer announced that they had invented a drug, and at its core is a protease. So therapies that can use targeted proteases are an interesting aspect. Okay. So the next class are DNAligases, and so if you recall, what do ligases do? They join monomer, nucleotide monomer, to the growing DNA polymer, right? Used to join DNA fragments. So techniques are used to add things to the ends of nucleic acid sequences, linkers or adapters, and you do this first by generating what's called a blunt cut, so that you don't have an overhang, so that the DNA is just reaches a simple endpoint, and then you can add things to the end of that. Okay, so obviously you can either join a single DNA molecule, or you can add two different molecules together. Basic genetic engineering, right? Okay. So the other class of enzymes are the so-called modification enzymes that do add things to the ends of DNA sequences. Terminal deoxynucleotidal transferase is an example. Okay. And then finally, restriction enzymes. So these are endonucleases. They're found in bacteria, and species of bacteria differ for the kinds of endonucleases they have. And what these enzymes do is they recognize certain sequences. The sequence may be only 4 bases long or 6 bases, etc. And wherever that message is, the enzyme, being an endonuclease, will cut the DNA at that point. So it became very obvious that this was an enormously important tool for manipulating DNA sequences. So even though the genome is really, really large, you know that if you can extract the DNA and you apply this endonuclease, you know that the fragments have been cut at the target site that you want. And so how you would choose an endonuclease is based on the sequence it has, certainly, but how long the sequence is would then generate how often you expect to see that message within the genome. So we've already used this equation before. So again, 4 different nucleotides, a message of 4 bases long. How often would you find that expected one? It's about basically every 256 bases. 6 cutter every 4,000 bases, and finally an 8 cutter about every 65,000 bases. So you can then already see that with a small genome, you might want to use a 4 cutter, but with a larger genome, if you're just trying to generate large fragments, that the 8 cutter might be a more appropriate sequence. Okay, so your book does do some justice explaining how you can plan to use different restriction enzymes, and so we'll see if my little animation goes, but you'll see, you know what a palindrome is? Okay, well the idea is that forward and backward the message is the reverse of it, okay? And so EcoR1 is a very famous restriction enzyme, and so where the sequence it recognizes here, and it cuts between the G and the A. Between the G and the A. All right. Let's see. Yep, there it goes. Oh, that red blob, that's my enzyme. Pretty talented, aren't I? This is a wonderful animation. Disney's going to be hiring me any moment now. Okay, the enzyme's cut, and that's the result. And so these are what we call sticky ends, because there's an overhang here and an overhang here. Okay, so cut products can differ. Okay, and so we're going to stop there, And again, what I'm asking you to do, let's see if I have my slide. Okay, just, yeah, this is the restriction enzymes, the different kinds of cuts, the sequences that are recognized, right? And so, where's our EcoR1, all right? I'm not going to ask you to sort of recognize these different sequences. We used to do that in the old days because we were really impressed by ourselves if we could remember these facts. It's called databases and computers. So in lab, for example, we're going to be using a genome workbench called Unipro Eugene. Okay, and so some of you have already started to download and install that software onto your computer. Good for you. We're going to use that throughout the semester to interrogate genome sequences. And so my point being is that Eugene contains all of the known restriction enzymes, and so you can then just go click and select, I want to know if there are any EcoR1 sites available in this DNA sequence that I have. Click, and it will find those for you, right? You don't have to memorize the recognition site. It'll generate those for you. That's what computers do, and thank goodness for that. Okay, so this is the slide I wanted to end on. Just as a reminder, there are 4 classes of enzymes that are talked about in the chapter. And I want you to try to think about the targets that they work with, the substrates that are necessary, how they work, so tell me a little bit about their activity, and then what's the end product, right? Okay, so I'm going to stop there and kind of race through this a little bit. And stay tuned to Canvas for information about Friday. I'm looking at probably producing a video and then getting it published for you, but there probably won't be any live stream. All right, and then the other reminder, of course, is that remember you're not doing lab today. So I'm busy behind the scenes, finishing up the sequence of projects for the lab. And so, have a nice day.